What if instead of endlessly searching for the perfect weakness in a solid tumor, we could just, hack into the cancer cells and force them to paint a giant bullseye on their own backs?
It sounds a bit like science fiction. But a brand new dual-key system using low oxygen and focused ultrasound is doing exactly that. It is tricking tumors into revealing themselves to our immune system, which completely changes the rules of engagement.
Welcome to the deep dive. Today, we are unpacking some truly groundbreaking peer-reviewed research published just yesterday, August 16th, 2026.
Right, in the journal Science Advances, by researchers at the USC Viterbi School of Engineering.
Exactly. And our mission today is to explore what might be, honestly, the most significant paradigm shift in cancer treatment we have seen in years.
It really is a massive leap.
So if you are listening to this and wondering why we have not already cured all cancers with immunotherapy, you are definitely not alone. To understand exactly why this USC study is so revolutionary, we first need to look at the immense frustration surrounding current cancer treatments.
We have to talk about the brick wall that researchers have been banging their heads against for the last decade.
Right. And that brick wall is the solid tumor, is it not?
It is. For over 10 years, a treatment called CAR-RT cell therapy has been nothing short of a miraculous breakthrough, but almost exclusively for blood cancers.
Things like leukemia and lymphoma.
Exactly. In those cases, physicians take a patient's own T cells, engineer them in a lab to recognize a specific marker on the cancer cells, and then infuse them back into the body.
So they essentially become a living, circulating drug.
Right. And the reason this works so beautifully in the blood is that blood cancer cells float freely. And they often carry a very specific uniform marker, like a flag that normal essential organs just do not have.
So the engineered immune cells simply swim through the bloodstream, spot the flag, and eliminate the target.
Precisely. But solid tumors, things like brain, breast, and liver cancers, play an entirely different game.
Because they are structured differently.
Right. If you visualize a solid tumor, do not picture a neat little ball of uniform cells. Picture a fortified, chaotic fortress. The environment inside a solid tumor is actively hostile to the immune system.
It is, it is dense, it is fibrotic.
Yes, and most importantly, solid tumors hide in plain sight because they lack those unique surface markers.
So if you try to program a T cell to attack a marker you found on, say, a breast cancer cell, there is a very high probability that same marker exists on healthy breast tissue.
Or in the lungs, or the liver. And if the immune system is unleashed on that marker, it does not distinguish between the tumor and the vital organ.
Which results in massive, sometimes fatal collateral damage.
Exactly. The scientific community calls this the needle in a haystack problem, or the search for the perfect antigen.
Because for decades, we have spent billions of dollars sequencing tumors, right? Trying to find one unique protein structure that exists exclusively on the cancer and absolutely nowhere else in the human body.
Right. And what the USC Viterbi team realized is that this search might be fundamentally flawed. Instead of looking for a natural marker that probably does not even exist, they engineered a system to force the tumor to manufacture a temporary marker on command.
Okay, let us unpack this because this is where it gets really interesting. We are going to explore this revolutionary new solution called shifters.
I love that acronym. It stands for Solid Tumor Hallmark Inducible Focused Ultrasound Triggered Enhanced Reprogramming System.
It's just a mouthful, but to strip away the acronym, it is basically a biological AND gate.
Exactly. If you are familiar with computer programming, you know exactly what an AND gate is. A certain outcome only happens if condition A and condition B are met simultaneously.
Right. If only condition A is met, nothing happens.
And if only condition B is met, nothing happens. The brilliance of shifters is that it applies this strict binary logic to living biology inside the human body.
So the researchers designed a genetic circuit that requires two distinct physiological triggers to activate. Let us break down the exact methodology here. The first key in this biological AND gate is a natural tumor hallmark hypoxia, which is a state of severe oxygen deprivation.
Right. And to understand why tumors are starved of oxygen, you have to look at how they grow. Solid tumors expand so aggressively that their cellular division drastically outstrips their blood supply.
Because normal tissues in your body have this beautifully ordered, highly efficient network of capillaries delivering oxygen.
Exactly. Tumors, on the other hand, frantically build chaotic, leaky day-end blood vessels.
So because this infrastructure is so terrible, the core of a solid tumor is basically suffocating.
Yes. While normal healthy tissue sits at relatively high oxygen levels, these deep tumor pockets drop down to around 1% oxygen.
Wow, just 1%.
Right, and the researchers hijacked this natural biological phenomenon to act as the first key.
Okay, so that is key one. Now, the second key is entirely external, right? And
Yeah.
completely under the physician's control.
Yes, it is focused ultrasound, or FUS.
Mm-hmm.
Most people associate ultrasound with imaging, like looking at a baby during pregnancy. But if you adjust the frequency and focus the sound waves using an acoustic lens, you can safely pass those waves through healthy tissue and have them converge on a single millimeter precise point deep inside the body.
And when those sound waves converge, they generate friction and heat.
Exactly. It is a non-invasive hyperthermia technique. The physician uses this focused ultrasound to heat the specific tumor tissue to exactly 43 degrees Celsius for 15 minutes.
And we should explain exactly what happens to a cell when it hits 43 degrees Celsius, which is about 109 degrees Fahrenheit.
Right, it gets very hot for a cell. When a cell experiences that temperature, its internal proteins begin to misfold.
So the cell realizes it is under extreme thermal stress, and to survive, it basically panics.
Yes, it activates emergency response mechanisms, specifically turning on heat shock genes to protect itself. And the shifter system is engineered to listen for that exact molecular panic.
So when both of these keys turn simultaneously, when the cell is experiencing the 1% oxygen environment of the tumor core, and it experiences the exact heat shock triggered by the focused ultrasound, the genetic circuit unlocks.
Exactly. The tumor cell is hijacked by its newly introduced genetic instructions and is temporarily forced to produce a protein called CD19 on its surface.
And CD19 is the crucial payload here. The researchers did not invent a new marker. They used one we already know perfectly.
Right. CD19 is the exact marker found on B cells, which we have been successfully targeting with CAR T-cell therapy for blood cancers for a decade.
So we have mountains of clinical safety data on how immune cells hunt CD19.
We do. And the genius is that CD19 does not naturally belong in a brain tumor or a liver tumor. By forcing the solid tumor to display CD19, they are suddenly speaking a language our most advanced immunotherapies already understand perfectly.
That is brilliant. And the paper noted this marker sticks around for about a week, creating a highly controlled attack window for the immune system.
Yes. That temporal control, the 1-week window, is a profound safety feature. It gives the engineered CAR T cells enough time to infiltrate the tumor environment, mount a robust response, and clear the targeted cells.
But because the marker naturally fades as the cell recovers from the heat shock, it prevents the immune system from remaining in a state of perpetual hyper-aggressive activation.
Exactly. In traditional therapies, runaway immune activation can lead to severe side effects or long-term immune exhaustion, where the T cells simply burn out and stop fighting.
Wait, I am stuck on something regarding the first key. You mentioned low oxygen is a mandatory trigger, but do not tissues like the bone marrow or the rectal mucosa naturally experience very low oxygen levels?
Yes, sometimes they dip right into what we consider physiological hypoxia.
So how did the researchers prevent the system from accidentally triggering in healthy hypoxic tissues? I mean, would the system not just activate in a patient's bone marrow and start destroying perfectly healthy tissue?
That is precisely the hurdle they had to overcome. And it required some incredibly elegant genetic engineering to solve. To prevent the system from firing in the bone marrow, they built the circuit using something called a split Gal4-VP64 transcription factor.
Okay, that sounds complicated.
It is, but basically they separated the activation machinery into two distinct, useless halves.
Ah, I see. It is like a nuclear submarine launch sequence. One commander holds one key, the second commander holds the other, and they are standing on opposite sides of the room.
Exactly. One key alone cannot launch the missile. In this biological circuit, the hypoxia-responsive promoter, the part of the genetics listening for low oxygen, drives only one half of the system, the VP64 activator.
Right. And the heat shock promoter from the ultrasound drives the other half.
Yes, the Gal4 DNA binding domain. Only when both halves are manufactured by the cell at the exact same time do they physically link up to form the complete tool needed to turn on the CD19 beacon.
But the paper outlines that they ran into a serious problem here initially. They noticed some background leakage.
They did. Under hypoxia alone, without any ultrasound being applied at all, a small trickle of CD19 was still being produced by the cells.
Which is exactly what we do not want happening in the bone marrow. Why was it leaking?
The leakage occurred because biological systems are inherently noisy. Even in standard conditions, genes fluctuate. When they initially tested the split system, the genetic amplifier they used was simply too loud.
Ah, they were using 8 tandem repeats of a protein tag called sun tag, right?
Exactly, an 8-oxun tag. A tandem repeat acts like a biological volume knob. 8 repeats meant the signal was turned up to the maximum.
So even a tiny accidental flicker of the hypoxia gene in the bone marrow would get amplified so loudly that the cell would mistakenly produce the CD19 marker.
Yes. To fix that leakage, they had to systematically tune those genetic volume knobs down.
So they engineered the system, dropping the amplifier from an 8-exon tag down to just a 1-exon tag.
Right. And they also modified another part of the circuit called the upstream activating sequence. They truncated it from 5 response elements down to 2, and eventually tested various configurations until they settled on a highly specific 5-by-RE model.
It sounds like they spent an enormous amount of time searching for the perfect mathematical balance.
They really did. They had to find the exact threshold where the noise in naturally hypoxic healthy tissues remained completely silent, but a strong, unmistakable CD19 signal fired the moment the heat shock condition was introduced.
And the precision of that tuning is the only reason the shifter's platform is viable.
Absolutely. By reducing the amplification, they established a strict floor. The baseline hypoxia found in a healthy organ will never be enough to trip the circuit on its own.
It absolutely requires the massive concentrated thermal energy spike of the focused ultrasound to push the activation over the threshold.
Right.
Here's where it gets really interesting, though. Once you have this targeted CD19 expression working perfectly, you immediately run into another classic solid tumor nightmare, which is heterogeneity.
Yes, tumor heterogeneity is a massive problem.
Because not every cancer cell in a solid mass is identical. And more importantly, an ultrasound beam is not going to heat every single cell perfectly evenly.
Exactly. Some cells will not get hot enough. Some cells might not be hypoxic enough.
So if we are relying on this system to paint a bullseye on the tumor, what happens to the cells that do not get painted? Do they just survive and regrow the tumor?
Well, the researchers set up 2D and 3D spheroid experiments using U251 glioblastoma cells, which are brain cancer cells, to test this exact scenario.
And this is where the research moves from a clever engineering trick to a potential clinical miracle.
It really is. They mix their engineered shifter cells with wild-type, unengineered tumor cells. So they essentially created a model where only a fraction of the tumor could possibly display the CD19 marker.
Right. And then they introduced a highly specialized immune cell called a CD19 Synach CAR T cell.
And the findings from those 3D spheroid models were astonishing. You do not need every single tumor cell to display the CD19 marker to destroy the entire tumor.
The paper states that if just 10% to 25% of the tumor cells are activated by the ultrasound to show CD19, the entire mass is still completely eradicated.
Yes, because the activated cells act as localized training centers for the immune system.
We need to explore exactly how a Synach cell achieves this because it is fascinating. A Synach receptor is not a weapon itself, is it? It is more like a biological tripwire.
That is exactly right. Normal SAR T cells are designed to immediately kill whatever they bind to. A Synach CAR T cell is different. When its receptor binds to the CD19 marker on that small percentage of heated tumor cells, it does not just attack.
Which is due.
The binding physically snaps off a piece of the receptor on the inside of the T cell. That snapped-off piece travels directly into the T cell's nucleus and acts as a specialized instruction manual.
Telling the T cell to start manufacturing a secondary weapon.
Yes. It is essentially a reverse zombie scenario.
A reverse zombie scenario.
Think about it. In a normal zombie movie, one zombie bites a human, and the infection spreads until everyone is a zombie. Here, one tagged cancer cell is found by a circulating immune cell, but instead of the cancer cell infecting the immune cell, the cancer cell triggers the immune cell to arm itself to the teeth.
Ah, I see. The Synach mechanism forces the T cell to locally manufacture and display an entirely new receptor on its surface, an EGFR CAR.
Exactly. And EGFR is a natural protein that happens to be highly overexpressed on the vast majority of those unengineered glioblastoma cells, the ones that never receive the ultrasound heat shock.
Oh, wow. So once armed with the EGFR CAR, the T cell undergoes a profound behavioral shift.
It does. It begins executing a localized slaughter of everything in the immediate vicinity, carrying that EGFR marker.
So the 10 to 25% of cells that displayed the initial CD19 marker served solely as a beacon.
Yes, they drew the resting T cells deep into the hostile tumor microenvironment, and upon contact, triggered the on-site manufacturing of the weapons needed to kill the rest of the tumor.
That is incredible. This bystander killing effect completely circumvents the limitation of tumor heterogeneity.
It really does. If a treatment only kills the 20% of a tumor that it can properly identify, you leave behind 80% of the cancer.
And that remaining 80% is often the most resilient, treatment-resistant fraction. It will rapidly mutate and grow back, usually much more aggressively.
Exactly. By utilizing the training center effect, the researchers guarantee that the initial imperfect targeting still results in total regional clearance.
When we look at the broader implications of this research, it fundamentally flips our entire approach to solid tumors. We noted earlier that for decades, scientists have fruitlessly searched for the perfect solid tumor antigen.
Right, and this technology suggests that the search itself is obsolete. We can stop looking for the biological lottery ticket.
Because instead of hoping a tumor happens to present a safe target, we simply dictate the target.
Exactly. It transitions cancer immunotherapy from a problem of biological discovery into a problem of mechanical engineering.
And the researchers also highlighted the extreme modularity of the shifters system. They did not just design this to work with hypoxia.
No, they wanted to prove it is a flexible platform. They went into the genetic circuit and successfully swapped out the hypoxia-responsive key for a different trigger entirely.
A liver cancer-specific promoter called an AFP, right?
Yes. They then tested this new variation on Hep3B liver cancer cells. The NFP promoter is highly active in a hepatocellular carcinoma, but it remains completely silent in healthy adult liver tissue.
So this modularity proves that Shifters is a highly customizable plug-and-play platform.
It is. If you, as a patient, have a tumor that is not particularly hypoxic, but has a different known biological hallmark, a doctor could tailor the genetic circuit specifically for your cancer.
They could swap in a breast cancer-specific promoter or a prostate cancer-specific promoter and combine it with the focused ultrasound to achieve that same pinpoint accuracy.
Exactly. The therapeutic architecture remains exactly the same. You just change out the sensor.
We must also examine the study's constraints, though, to keep this excitement firmly anchored in scientific reality.
Right. We have to look at the limitations.
The most robust tests they ran were in vivo, meaning live animal models. They utilized a bilateral flank model in mice.
The experimental design there was quite elegant, actually. They implanted human tumor cells on both the left and right flanks of the mouse.
However, they only applied the focused ultrasound to the tumor on one side.
Right. After the ultrasound, they injected the mice with a circulating dose of the engineered T cells, escalating the doses from 3 million up to 10 million cells.
Now, the bilateral flank model is a rigorous, standard way to prove spatial control in immunotherapy.
It is, because the T cells are injected into the bloodstream, so they circulate systemically throughout the entire body of the mouse. They have equal access to both tumors.
But the researchers observed that only the ultrasound-treated tumors shrank dramatically and were cleared.
Yes, the untreated tumors on the opposite flank of the exact same mouse continued to grow aggressively.
Which proves unequivocally that the T cells only activated and engaged where the ultrasound heat shock turned the system on.
Exactly. But we have to state clearly the caveat of using these specific mice.
Right. The mice used in the study are called NSG mice, which means they are deeply genetically immunocompromised.
They basically have no functioning immune system of their own.
And this is a requirement when you are implanting human cancer cells and human T cells into an animal, right? Otherwise, the mouse's body would immediately reject the foreign human tissue.
That is correct. But because the mouse has no immune system, we are not seeing how a fully intact human immune system might interact with this process.
Because a real patient's immune system might recognize the engineered shifter cells as foreign and destroy them before the ultrasound ever happens.
Or it might create massive inflammation that suppresses the CAR T cells. We just do not know yet.
And alongside the complexities of a real human immune system, there is a massive delivery hurdle that the paper acknowledges.
Yes, delivery is a huge challenge.
Because the technology, as demonstrated, relies entirely on pre-engineered cell lines.
Right. The researchers took the cancer cells, altered their genetics in a petri dish, using sophisticated lab equipment to insert the shifter circuit, and then implanted those pre-hacked cells into the mice.
But in a clinical setting with a human patient, the tumor is already inside the body, growing naturally.
Exactly. Doctors will need a practical, efficient way to deliver the shifter's genetic program directly into an established, hostile solid tumor.
And the delivery of genetic medicine is arguably the hardest problem in modern biotechnology.
It really is. The researchers noted they're currently comparing delivery methods for future clinical use. Specifically, they are looking at lipid nanoparticles versus modified viral vectors.
Lipid nanoparticles, or LNPs, are the tiny fat bubbles used to deliver the mRNA vaccines, right?
Yes, they are safe and easy to manufacture, but getting them to penetrate deep into a dense, fibrotic solid tumor is incredibly difficult. They tend to just bounce off the dense outer layers.
Ah, I see. So what about the modified viruses?
Well, modified viruses like lentiviruses or adeno-associated viruses are exceptionally good at infecting cells and inserting genetic code.
But using viruses carries its own risks, I imagine.
It does. Things like triggering an unwanted immune response against the virus itself, or the complexity of manufacturing enough clinical-grade virus for thousands of patients.
So developing those specific delivery vehicles, conducting the much larger animal studies in primates, and progressing to phase 1 human trials means this technology is still years away from the clinic.
And it will require massive financial investment to progress.
Let us look at the expert consensus regarding safety, though. If we connect this to the bigger picture, the scientific community's hesitation with using CAR-T in solid tumors has always centered on off-tumor toxicity.
Right. The very real risk of highly engineered immune cells going rogue and attacking vital organs, which can be fatal in a matter of days.
But the true genius of the Synach prime and kill paradigm used in shifters is its built-in failsafe mechanism for the T cells themselves.
Exactly. We mentioned earlier that the T cells manufacture a secondary weapon, the EGFR CAR, once they enter the training center.
Right.
What makes this exceptionally safe is that the EGFR weapon is transient. The genetic instructions degrade quickly. In fact, the secondary ERR expression has an 8-hour half-life.
Wow, just 8 hours.
Yes. So when the T cells eventually run out of targets in the tumor and inevitably leave the ultrasound prime zone, migrating back into healthy circulating tissue, they physically lose their secondary weapons.
The CRR expression just wanes completely.
It does. This means doctors can potentially use incredibly potent, highly lethal immune attacks within the tumor microenvironment without the fear of those immune cells ravaging the rest of the patient's body hours later.
That is a massive relief for clinical safety. Well, it is time for our signature segment, Myth versus Fact.
I will take the myth this time. The myth we are busting today is the long-held belief in oncology that to successfully cure a solid tumor with targeted immunotherapy, you have to find a way to tag absolutely every single cancer cell in the mass.
And the fact, proven beautifully by the 3D spheroid models and the live animal tests in this paper, is the power of the training center effect. You do not need to tag every cell.
Not at all.
Tagging just 10% to 25% of the cells creates enough localized, intense immune activation to arm the circulating T cells and wipe out the entire tumor neighborhood. You only need to paint a target on a tiny fraction of the cells to achieve complete eradication of the whole mass.
Right, exactly.
So what does this all mean for the future of medicine? We have successfully engineered a remote-controlled biological switch that uses pinpoint-focused ultrasound and the natural low oxygen of a tumor to force solid cancers to paint a target on themselves, making them highly vulnerable to our best CAR T-cell therapies.
And if we can use an AND gate genetic circuit to make tumors reveal themselves safely, imagine the future applications across all fields of medicine.
Right, it goes beyond cancer.
Could we use this exact same modular ultrasound and gene logic to temporarily target and clear out amyloid plaques in the brain for a patient suffering from Alzheimer's disease?
Or could we swap the genetic keys to target and clear out fibrotic scar tissue in the heart after a patient suffers a severe heart attack?
The potential for remote-controlled, precisely targeted cellular medicine extends far beyond oncology.
It really is incredible to think about. If you want more science-based health breakdowns that dive deep into the research, subscribe for next week's Deep Dive.
Thank you for joining us today. We will see you next time. And remember, sometimes the best way to hit a hidden target is not to build a better arrow, but to force the target to paint a giant bullseye on its own back.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.